A major part of our recent focus has been next-generation sequencing and supporting all of the next-gen platforms, technologies, and workflows. However, it really is only fitting that our first webinar here focus on Lasergene Core Suite, which we've been building on for almost 25 years and includes traditional molecular biology needs like primer design, virtual cloning, sequence editing, and sequence alignment. We pride ourselves on making software that's easy to use, but we also know that issues can and do arise over time where our customers will need our help. So in addition to building good product, we have put a strong focus on providing effective, timely support. We offer free trials on every page of our website in the upper right corner. And we also have tutorials for major workflows. We have FAQs, and of course, we offer live technical support. You simply just need to email us or call us for a fast response by one of our scientists. About 18 months ago, we began a publication called the Lasergene Weekly. It highlights what's new in Lasergene and with our company, but more importantly, each week we provide a 3 or 4 minute video on one of our key software workflows. We have about 100 videos in our video library right now. You can watch them from the web page we're scrolling through, or you can watch them on YouTube if you prefer. We have our own channel there. And we add about 1 or 2 videos every week. If you aren't sure how to accomplish something using our software, see if we've already got a video on your topic of interest. If not, let us know, and we'll add it to the list of future video topics. Which leads me to why we've initiated this series of webinars. Different people learn in different ways. We want to support as many ways of learning as possible. And when added to all the other ways we provide support, which I've tried to briefly walk you through here, we believe webinars represent another way for us to provide value to the scientific community by making it as easy as possible for scientists to use our software to successfully accomplish their research objectives. So without further ado, on behalf of all of us here at DNASTAR, I want to thank you for choosing to spend some time with us today. And I'm going to turn it over to Ryan Jacob, who has been supporting people like you using DNASTAR software for more than 10 years. Ryan's going to give an overview of some key functionality of DNASTAR's Lasergene Core Suite, and then he'll do a deep dive into a couple of our more popular applications and workflows. And as Katie said, we have ample time in here for questions during and after Ryan's talk, so please feel free to submit any questions you have using chat anytime during the webinar. Ryan?
Thank you, Tom. So my name is Ryan Jacob. I've been here, as Tom mentioned, for over 10 years in a sales and technical support role. So today my main goal is really to take you through some of the different Lasergene applications that we have in the Lasergene Core Suite. And just give you, I'm going to start with a brief 5 to 10 minute PowerPoint presentation just so I can introduce all the different applications in the DNASTAR Lasergene Core Suite. And then I'll go into 3 of the applications in more detail and actually go into the software and show some major workflows and just give you some examples of real data and so forth. So just to start out here, this is what's called our Lasergene Core Suite. This is the DNASTAR Navigator, and this is basically a launching pad for some of the different applications that we have. You can see here on the very top is the DNA sequence analysis package where you can load in DNA files to these 4 applications. Next we have the proteomics package where you can focus on loading in protein sequences, and then some variety of utilities that we have as well. So I just want to talk real briefly about each one of these in particular. The first 3 here, the SeqBuilder, MegAlign, and SeqMan Pro, I'll be going into that software in a bit more detail, but I just want to do a brief introduction in this PowerPoint first. So as I said, the DNASTAR Navigator is really a launching pad for a lot of the major applications that we have. You can just launch this in the background and simply click on one of the applications to launch that particular tool. But it's actually much more than that as well. This is just giving you a screenshot. If you hover over the SeqBuilder application, you can get a feel for what the SeqBuilder provides for you, the functionality. You can get some training videos, tutorials, and download example data as well. So any of these links will actually take you to our website where you can learn more about the actual particular application. Beyond that, you can actually connect with the help files here in the help menu. So if you click on this blue little eye palette tool at the top, that will basically allow you to get access to the entire help manual built into all of our tools. And it's really, really nice where you can search the functionality of that help menu for a particular workflow that you're looking for, or just a particular keyword, and it will take you directly to that area where you can look at the parameters and any additional details that you want about that particular item. So just to talk about, introduce SeqBuilder very quickly here. SeqBuilder is really the main core module to the whole Lasergene Core Suite. What you're seeing here is a virtual clone. Basically, SeqBuilder allows you to import any sequence information, manipulate the annotations, do virtual cloning. Here you're looking at a linear map in the background and then a clone on the right-hand side showing you the annotations, open reading frames, restriction sites, and a lot more information. And I'll get into SeqBuilder in a few minutes here. Then we have our SeqMan Pro module. I'll also be talking about this module in a bit more detail. But the main purpose of this program is to assemble and analyze Sanger sequence data. In the background, what you're looking at is an assembled config, an aligned config with your consensus sequence at the top, your your translations at the bottom, and then in the middle section, you're looking at your chromatogram or your trace files of your actual sequence reads. And then on the left-hand side is your strategy view of how your sequences are aligned into that particular config. And then here's a quick image of SeqMan Pro again. This is just showing you an example of locating and filtering your SNPs. In the background, you're seeing your blue putative SNPs, and then in the bottom section is your SNP report of all your found SNPs along your config. And once again, I'll be talking about SeqMan Pro in further detail. Next, we have MegAlign, which is another multiple sequence alignment tool. This can be focused on either DNA or protein sequences. In the background, you're seeing a variety of different protein sequences aligned with a consensus sequence at the top, and then the bottom section is showing you our phylogenetic tree. Next, we have our GeneQuest module. GeneQuest is a really great module for loading in a DNA template file and looking for different patterns and searching for different genes of interest. You can do regulatory elements, predicting coding regions. You can see the assay surface in the middle of the screen showing you a different variety of methods that have been applied to your actual DNA sequence. On the right-hand side is a legend showing you the open reading frames, Borodowski's matrices, and the different methods that you can apply. And then on the left-hand side are these particular methods that have been applied to that particular DNA template sequence. Next, we have Protean 3D. Protean 3D is a great program to use that was introduced just a few versions ago. Basically, if you work with protein sequences files and want to look at the 3D structures or apply different methods to your protein sequence and look for secondary structure and other types of things, this is really a great program to use. It's very interactive, so you can actually look at your protein 3D file at the same time as actually looking at your annotations and looking at your actual amino acid sequence on the left-hand side here. Next, we have one of the utilities, GenVision. Basically, this is a great program to use, especially if you've sequenced a new genome and you want to publish that new genome, basically in a nice image, a nice image that is publication quality. And so it's giving you an overview of that particular genome. There's other applications as well, and it's a great program to use for those types of things. And then we have another new program that we introduced with the Lasergene 10 software that we came out with in March, SeqNinja, used for automated editing of sets and large sequences. You can see some of the applications here. Right now it's a script-driven program, but in the next major release of Lasergene 11, we'll have a graphical user interface built into the program, so it'll be quite easy to use once that comes out. And then this is just a screenshot showing you which modules I'm going to focus on in a bit more detail after this PowerPoint here. The SeqBuilder software, as I mentioned, SeqMan Pro and MegAlign. These are the three modules that I'll be focusing on and showing you some actual workflows within the software. So I'm going to end the PowerPoint presentation here and actually launch the SeqBuilder software and go into that in a bit more detail. If you have any questions after the end of each module, feel free to chat in the questions to Katie, and she can relay those questions to me, and you can ask a few questions at the end of each module that I present. If we don't get to your questions, no worries. We'll answer those after the webinar. We can send you an email or give you a call with more information if you have more questions. So if I launch the DNASTAR Navigator, this is what I was talking about in the beginning of the PowerPoint presentation. This is really a great launching pad for the software. So all I have to do is click on SeqBuilder, and it launches the program automatically for me. And I'll increase my screen size here. So I just want to talk about some of the different ways to import your sequence files into SeqBuilder. As I mentioned, this is really the core module to the whole suite of software. So when you're first importing data, chances are you'll use SeqBuilder to do that. If I go to the File menu option and choose New DNA, it just brings up a blank screen where I can simply type in my base pairs. You can copy and paste a file from Word or Notepad or any type of file that you have that you can copy and paste directly into this window. You can do that with new protein sequences as well. But the most common way to really get sequence files into the program is either by drag and drop or by going to the File menu option and choosing Open. And at the bottom you can see the different file types that we can open into the program. So a lot of the main DNASTAR sequence files you can open. Beyond that, we can open up GenBank files, FASTA files, and a variety of other files that are listed here. So I'm going to go to one of my example sequence files and open this up into the program. And you can see what's opened up automatically is what's called our sequence view. On the left-hand side, you have the different windows and file folders that you can control a lot of what you see on the right-hand side. You can see that the sequence view is shown just because it's blown up a little bit. It's a little bit more enlarged versus the other items that we have here. You can see the 5' end of the sequence and the 3' end with a scale, your translation underneath that, features and annotations, and your restriction sites as well. All of these things are controlled on the left-hand side as well. So if I don't want to see my features, for example, I can uncheck my features, and that will basically just hide the features and annotations that are directly on my sequence. As I scroll through the different sequence views, I can show you a few examples of the other types of views. Under the feature list view, this is just all the annotations created with my file. If you download a GenBank sequence file from NCBI, all the features will be in GenBank feature format. Next, you have the comments view, and these are comments, once again, downloaded from a sequence from NCBI. Next, we have the linear map. This is a nice little overview of a linear sequence, showing you the annotations, the restriction sites, and a scale of basically how long that sequence is. You can zoom in and zoom out of this sequence as well, and it allows you to do a few other manipulations with it as well. Next, we have the circular map. You can zoom in and zoom out of the circular map as well, and there's other manipulations you can do with the actual annotations. You can move those around. You can move around the restriction sites, and you can change the size of the actual plasmid here as well. Next, we have the primer design view. This I'll get into a few examples in a moment, and I'll show you an example of this primer designing. And then the primer list view as well. Once you find primers on your sequence, it will show you this in a list format here. Next, we have the minimap view, and this is basically just showing you the restriction sites that are applied to your particular sequence, the frequency that they cut, and then these little blue hash marks on where they're cutting on your sequence. And then you have the site summary view showing you another list of the restriction sites, the frequency that they occur, and the position that they occur on. And this is a sortable list. If you click on any of the headers at the top, you can resort those based on the frequency or the actual name of the restriction site. So if I go back to my sequence view, one of the nice things that you can do with the program is if I go to the window menu option and choose horizontal split, you can actually split the screen in two so that you can look at two different views at the same time. And why this is useful is that you can see all the features at the bottom. You can double click on one of the features and that automatically highlights it in the top sequence view. And you can do that with any of the different views. So you can show any of the two views together simultaneously. And you can even split your screen multiple times if you have a big enough real estate to work with on your screen. I'm going to minimize this just for ease of use here. On the left-hand side, I already showed you some of the features that you can apply. We have the features displayed by alphabetical list, positional list, or feature type. So you can just singly click on one of the features to apply that feature, make that present on the right-hand side, or make that go away. To create a new feature, if I uncheck my features here just for simplicity, if I highlight a region and go to the feature menu option and choose new feature, it automatically creates a red arrow for me, which you can change as what you would want for the default color and the style. I can change this if I just right-click and say Edit Feature Description. I can type in my new feature and apply that to the actual screen. Double-click on that feature and you can see my new feature label on the inside. You can also change the color of that feature. If I change the edit feature style, you can go to the palette tool and change the color of that feature, or even the style, whether you wanted an arrow or a box or another type of shape that you would want in for that annotation. You can see the color has changed, and it changed the style as well. One of the things I wanted to show you as well is the enzyme selectors and some of the enzymes that we have on the sequence. To do this, I'm just going to jump over to the circular map. I'll show our features as well. You can see a variety of different enzymes here shown on your plasmid. On the left-hand side, you have an enzymes displayed. There's two ways to display your enzymes, by our built-in selectors or just an alphabetical list of the actual enzymes. So you can click on the particular enzyme that you want, but an easier way to do this is really by these built-in selectors. And these come installed with the program with a variety of different selectors, but you can change any of the selectors and create new ones as well. And to do that, you just go to the Enzymes menu option and choose Selector Manager. I'm going to delete this so I can show a new one here. And if you click on New, you can just give this a label like Unique Sites. And I'm going to search for restriction sites that are a frequency type of selector that cut just one time on my sequence. So if I click OK and then go to the left-hand side where the filters by selectors are and click on Unique Sites, you get a list of just the restriction sites that occur one time on your sequence. And you can verify this by going to the top and going to the Site Summary view that I showed you before, and you get a full list of all these restriction sites, and the frequency is just one time on the sequence. And one of the nice things about this is that you can show with your selectors, basically you can combine that with one another as well. So if you want to order through the company ProMega and you want to look at unique sites, that will basically filter through that and just show you those particular restriction sites that you can order through the ProMega company. A couple of the other things I wanted to show you before I moved on to my next workflow. Basically, there is a blast menu option and a net search menu option located under the net search menu at the top. And this is built into all most of the software modules. It's not built into Protean 3D, but all the other modules within the Lasergene Core Suite have this application. And you can highlight a region of sequence and do a blast search, or you can just do a new text search for a particular author or a particular accession number that you have. And you can download the sequences from NCBI. I'll just show you a quick example of this by searching for Fred Blattner. He's the CEO and founder of our company. And if you get the results here, you can easily click on one of the results and click on Create Document, and that will actually load that particular document into the SeqBuilder software. It will give you all the annotations from NCBI and all of the correct sequence information directly downloaded from NCBI. One of the other features I wanted to show you as well before I moved on, under the File menu option, you can send the sequence to any of the other modules directly in the Lasergene Core Suite. If you just go to Send Sequence To, I can click on MegAlign, and it will automatically launch that sequence directly in the MegAlign software. And this is very nice for actually editing the sequence. For example, if I want to edit the sequence in SeqBuilder, I can go to the front of my sequence, the very beginning, and type a variety of different A's here. And then you can go back to MegAlign, and you can see that on the fly, it's actually updating that program, that sequence, while you make edits in the SeqBuilder software. So the programs are interactive with one another, and you can send the sequence files from one program to the other, and really get a good use out of your sequence for the different workflows that you might have. So I'll move on to one of the other projects that I wanted to show you. I want to go through a cloning example, so I'm just going to close down some of these windows that I have open here. And I'll show you a restriction clone and the way that we work our cloning projects, our virtual clones. If I go to the File menu option and choose New Cloning Project, you can see that it opens up a new window with my inserts, my vectors, and my clones. And this is a nice way to keep track of these different parts of your cloning project. If I right-click on my Inserts folder and choose Import, I'm going to go to some of my demo data and import one of my sequences here. And you can see that's imported into the Inserts folder. One other thing I'm going to do is go to File, Open, and go to my Cloning Vectors file. Now this is just a database of all the vectors that we have built into the software, and this comes installed with the software when you first install it. I'll go to my restriction vectors and locate one of my more popular restriction vectors that I have. And I'll just right-click and choose Copy. And I'm going to actually paste that into my vector folder in my Untitled Cloning project. And you can see it's just copied over from my database into my new vector. You can add any new vectors that you work with on your own to this vector file, this vector database that you have, and you can work with those in the future for future projects. And then from here, I'm just going to double-click on my insert file, and that's automatically going to load that sequence file into SeqBuilder. And you can see it's in the linear view at this particular time. If I go to my enzymes displayed, I'm going to show the selectors that I created before, and then click on my unique sites. If I click on my first restriction site, and then hold my Shift key down and click on the last one, it will actually highlight the whole sequence range in between those two restriction sites. And then from the cloning menu option, I'll choose Copy Restriction Insert. And then if I double-click on my vector file, I will choose my selector file again and select my unique sites. And then on the right-hand side, you can see, if I zoom in a little bit, and then I'll click on the same two restriction sites that I had before, and highlight that range of the sequence. And then from the cloning menu option, I'll choose Clone Restriction Insert. And this is really where the power of the program comes in. It's going to basically tell you whether you're matching up the sequence ends. Basically here I chose the same two restriction sites, so you can see the left and the right ends are matched up with this check mark. If I move these arrows at the bottom though, you can see that the check mark disappears and the clone menu option is not active any longer. So it's basically allowing me to match up my ends before I actually perform my clone. And then if I click on my clone menu, you can see that it performs the clone, and it creates the new feature on the actual vector where my insert file was. If I minimize this, you can see from my Untitled Cloning project, I have my insert, my vector, and my clone all within one project. And then from here, I can choose File, Save As, Save Project As, and it will actually save it as a SeqBuilder project file, an SBP file. And so when I open up this project in the future, I'll always be able to keep track of these inserts, vectors, and clones that I used for this particular cloning project. One other important thing to point out is under the cloning menu option, you have a variety of different cloning tools. You have TA cloning, gateway cloning, both multi-site and regular gateway, and then directional topo cloning as well. And these are very easy to use cloning, virtual cloning methods built into the software, so I encourage you to look at those in the future on your own as well. So I'll quick close down this program and then show you one other example in SeqBuilder, and that's our primer designing example. If I go to File, Open, I will work with one of the sequence files that I had open before. Double-click on that file. And in this particular case, I'm just going to actually click on one of my features in my particular linear map that I have. And then from the priming menu option, if I click on Create Primer Pairs, it will actually bring up a dialog whether I want to create my primers exactly at my selection or choose an optimal primer length within a certain number of base pairs of that selection. And then it will give you a variety of different conditions and parameters at the bottom and characteristics that you can change. For example, if I want to change my melting temperature to be closer to 55 degrees Celsius, I can change that here. If I click on OK, that automatically creates my primers. My upper primer is in green, and then my lower primer is in red. And at the top, you can see your actual primer sequence. At the very, very top, you have your length of your primer, your melting temperature, and you have these two little arrows between your actual primer. And you can click on these arrows to slide this over, and it's changing the actual length and the melting temperature as you do that. And it has a variety of other windows here at the bottom. It's going to show you whether your primers are stable, whether you have any hairpins and so forth at the actual primer. And then your primer list that I showed you before, you have the set one that was developed, and it will actually show you your primer sequence here as well. So there's a lot of other tools that we have for the actual primer designing and the cloning options, but I'll end the presentation, at least for SeqBuilder at this time, and I'll open it up to a few questions as well, and then I'll move on to the SeqMan and the MegAlign software. Okay, well, there's no questions at this time, but I encourage you to email us in if you have any questions. You can just email info@dnastar.com or give us a call toll-free at the numbers listed on our website as well, and we can offer any advice on your actual projects or answer any questions further. So I'm going to close down this SeqBuilder software, and I'm going to move on to the SeqMan Pro software just simply by clicking on SeqMan Pro in the DNASTAR Navigator. And as I mentioned earlier, SeqMan is really a program for working with your Sanger sequence data and doing things, aligning that data into contigs, looking for SNPs, doing things like primer walking, just a lot of different tools built into SeqMan. It really is one of the most powerful modules that we have. So this is the first screen that you look at when you actually launch SeqMan. You'll see your unassembled sequence window that appears. And the easiest way to get started with the program is click on this Add Sequences button. And what you'll do is search for some demo data and load the data into the program to get started. And I'm just going to load 14 sample ABI files that we have. I'll click on Add and then Done. And those are automatically loaded into the unassembled sequence window. Now there's a variety of different things that we can do to the sequences before we actually perform the assembly. And that includes trimming the ends. If you want to trim the particular ends by a certain number of bases, you can do that here. You can mark a reference sequence to align these sequences to a reference, and I'll show you that in a minute. You can scan for a vector file. In this case, I'll click on No Vector here, and then click on Janus. I know the Janus vector is a part of these sequences, so I want to eliminate that before I do the assembly. And then a variety of other options that you can do here as well. You can trim your sequence ends and scan for vector, as I mentioned. Part of the nice thing about the program is it will trim the sequence ends automatically, and you can set a stringency to this actual sequence trimming that it performs. If I click on Scan All, you can see here the little check mark appear next to the Janus vector, meaning that that vector actually has been eliminated from these particular sequences. And then you can see the limits as well. You can see some of the sequences have been trimmed off. And I can verify that by going to Project Trim Report, and you can actually see a report of my actual particular sequences, the average quality, and the amount trimmed off of each sequence. So now I'm ready to assemble my sequences, and I'll click on this Assemble button in the top left-hand side of my unassembled sequence window. This is a report file. This is a handy file to look at. If you don't have sequences that were aligned into your config, it will tell you basically why those sequences weren't aligned. If the percentage of the match percentage didn't reach your certain threshold, or if you had too many gaps or so forth, it will tell you that information right here. One of the important areas to point out in the program, if some of these sequences didn't align into your config, you may want to adjust some of your parameters, and that's under Project Parameters. You can see here under the assembling parameter, for example, you can see these percent matches were all in the 90s and 100s. If it fell below 80%, basically those sequences would not have been aligned into the config, and you'd have to lower that minimum match percentage if you did want those sequences aligned. Another popular parameter to switch is the minimum sequence length. If it falls below 100 base pairs, then it won't be assembled into these contigs either. And there's a large variety of different parameters to adjust here, and you can go through some of these on your own. One of the nice tools that's built into each of the applications is the Help menu. There's a shortcut to the Help menu here if I just click on the question mark, and it will automatically go to that particular window that I have open. In this case, it's the assembling parameters, and you can quickly see exactly what each parameter does and get a definition so you can adjust those in the future for each individual project. The other way to get to the actual Help menu is just under here, Help, Contents. You can get through the whole user's guide is built into this application here, and you can do a quick search for anything in particular, and it will automatically give you that information and those results, and you can quickly see those results there of the particular search term that you performed. So I just wanted to show you that. It's just a very powerful tool built into each software application, built into the help files. I'll go back to my actual sequence here. On the right-hand side, I have my untitled config, my config 1 that was formed. You can see the individual sequence files at the bottom that were assembled into that particular config. If there's more than one config assembled, it will be listed here as well, but I just have one in this particular scenario. If I double-click on the config, you'll see one of the most useful views in the program. This is the alignment of config view, and it's basically showing you your consensus sequence at the top and your other sequences that were assembled into your config on the left-hand side. You can scroll through that here and see your individual sequences. You can see the little arrows next to the left or the right-hand side of your actual sequence, and this is the area that's been trimmed off of each sequence. One of the nice things about our program is you can click on that triangle, drag it to the left, and it will show you that trimmed data. So if you need that data back after the actual assembly, that particular data is not gone, you can rescue that data after you've done the assembly. And then on the left-hand side, you can see your actual sequence names with the black triangle. If you click on any individual black triangle, you will be able to see your chromatogram or your trace files if they were included with your actual sample data that you loaded into the software. You can quickly see all of them at one time if I just hold my Control and my Alt key down and click on one of the triangles, and it actually opens up all of these at the same time. If I click on this Q with the X through it, you can see, and then go to the actual Q, you can see that each trace file has an actual score assigned to it. One of the unique things about our program is that we assign a score to each individual base, and this just helps the consensus caller. So it helps make the correct consensus call at the very top. One of the other windows that's very useful directly in SeqMan is under the config menu, and you have strategy view. And this is what I showed you in the beginning on the PowerPoint slide as well. There's only 14 sequence files here. When you do have several hundred or several thousand sequence files, this window becomes a lot more important. You can tell exactly what direction the sequence files are pointing and how much coverage that you have in your actual config. And then this histogram at the top will actually show you the depth of coverage and whether it reached your threshold that you had set in your parameters. One of the other nice things about this report is that you can double-click on any area of your actual strategy view. If you have too thin of coverage, you can double-click on this area, and it will actually jump immediately to that alignment of config view. And that will allow you to basically go to that position and see if you want to sequence any more information. If there's any mutations or SNPs, you can look at that in this particular region as well. So I'll move on to one of the other workflows that I wanted to show you directly in SeqMan, and that's just going to be one of our SNP workflows, aligning some sequences to a reference file and basically showing you some of the different SNP tools that we have built into the program. So if I close down the windows that I'm working with now and go to the File menu option and choose New, you can click on Add Sequences again in the Unassembled Sequence window. And then I'll choose SNP Demo from the SNP folder that I have. And then I'll click on Add All and Done, and it loads 6 sample files along with a reference sequence that I have into the program. And this is just 3 individual sequence files, forward and reverse, labeled -F and -R for each sample. If I click on my reference sequence and highlight that, and then click on the Mark Ref button, you can see that it actually turns to an italicized font. And that means that all of these sequence sample files are going to be actually assembled to this reference sequence. If I click on the Assemble button on the right-hand side, you can see the config that's formed, along with the bottom section showing you the actual sequences in that particular config. Double-click on that config again, and this window is going to look a little bit different than you saw before. You have the reference sequence on the very top, as well as the consensus sequence. So these 2 sequences are exactly the same. It took these samples and aligned it to the reference file. If I click on the triangle next to the reference sequence, as I scroll through, you can see the different annotations that it had. And then you can actually hover over those annotations to learn more about those particular features that you have on your reference sequence. So when you do a reference-guided assembly, I would encourage you to use a reference sequence that you downloaded from NCBI or that you've annotated yourself. Because if they're GenBank annotations, you can see all those annotations directly in the software. When you're looking for variations in SNPs, you can particularly see whether there's a known variation or not, and other types of features like CDSs and exons across your reference sequence. So if you go to the snip menu option from here, and I choose Show Snips, basically as you scroll through your sequence, you can see that it grayed out a lot of the bases, and then it put in blue and green the most important areas that you want to focus on. And in blue are the any putative SNPs that you have, and green are the confirmed SNPs on the reference sequence. Another way to see this is go to the snip menu option and choose Snip Report. And this is basically a report of all your known SNPs on your particular config and the actual sequences as well. You have all found SNPs at the top, and then the SNP summary. I find the SNP summary view is I like the best, just because it's giving you each individual position on your config. It gives you a lot of information here: the reference position, the type of SNP that it is, whether it's an indel or an actual SNP, the reference base and the called base, and then the percentage of SNP that's occurring at that position. So for example, if you have 10 sequences deep and 5 out of the 10 sequences have a SNP, it would be a 50% SNP call in that particular location. It's connected to the DB SNP information, so if you work with an annotated reference file that has known DB SNP information, you can see that here. Your codon table, if your actual SNP is in a feature, it will show you that here. The coding distance from that particular feature as well. Just a lot of information in this particular SNP report. There's also a way to filter through the SNPs. Here I only have 10 or 15 SNPs, so it's pretty easy to see. But if you want to filter through and show me only SNPs that occur at least 50% of the time at each specific location, you can minimize the actual SNPs that you see in this report as well. And then a depth of coverage is also another easy way to filter through these SNPs. If you want at least 5 or 10 sequences deep, you can change that here, and that will eliminate some of the SNPs in your report as well. But the most handy feature really about the SNP report is that it's interactive with the actual alignment view. So I can click on the first SNP and use my arrow key and just scroll through each SNP, and that actually jumps to that particular alignment location. And I can look at the chromatogram files from here and verify that it's an actual SNP or not. You can see here in this particular location at position 58, you can see that it's 100% SNP call. All of the actual samples have this particular base called. If you click on the question mark on the left-hand side, just one time, it will actually turn to a check mark. And you can see that the SNP actually turns to a green color, meaning that you've confirmed that SNP, and it's now confirmed in your consensus sequence. And what it's doing is it's actually creating an annotation feature, variation feature, sorry. And when you automatically confirm that SNP, you can send that to SeqBuilder or other applications within the Lasergene Core Suite and see that variation feature along your consensus sequence. If you decline the SNP, you can look at one of the lower percentages SNPs and actually click on the question mark twice, so it turns to an X. And that actually turns your SNP to red, meaning you've rejected that as a possible SNP. So these SNP views are very interactive with one another. It's a very easy way to go through your assembly and really focus on the particular areas that you want in more detail. Now, if I close down this project, I am going to actually start fresh with that same workflow or those same sequences that I was working with. Click on Add Sequences and add all of those sequence files again. One of the nice features built into the software is a feature called Assemble in Groups. And basically, I just want to show you how we can work with that workflow in this software program. If I click on the reference sequence and mark ref again, it will make that reference.seq my reference sequence. And then if I click on the Assemble in Groups button, what this is doing, basically if you have, for example, a 96 well plate and you have a forward and reverse sequence in each 96 samples, and you want each 96 of these samples to go into a separate config, you can do that by the naming convention of your actual files. So what it's asking me to do is actually highlight the unique portion of my sequence name. So if I highlight sample 1, you can see that sample 1-F, sample 1-R are going into my sample 1 group. Sample 2 is going into the second one, and 3 into the third one. And then if I click on my Assemble button, you can see that it's assembling into 3 different configs this time, and 1, 2, and 3 here. You can see that just by double-clicking on the actual sample files, and these are individual contigs with that particular sequence read, those 2 particular sequence reads in each one. One of the nice things about the SNP report in this particular case is that you can actually show the SNP report for all the contigs at the same time. So you can see the contig ID, you have sample 1, sample 2, and sample 3. So if you're looking at a lot of data at the same time, a very large sample data set that you have, and you want to compare all the SNPs to one another, you can do that here in the SNP report versus all the contigs together. There's a lot of other features directly in SeqMan. I can't get to all of them today. Some of the other features that we do have under config, you do have primer walk. That's a nice feature if you want to actually design primers and extend the primer ends or walk into any of the gaps that you have along your sequence. And that's using that strategy view that I showed you before to find out where there's areas that you might want to improve the coverage on. That's a very nice, useful feature directly in the software. But you can experiment a bit on your own as well with the software and explore some of the features and some of the other videos that we have on our website. So again, I want to open it up to a few questions that you might have about the software.
Ryan, we do have a couple questions. If you have other questions, please chat them in. We can try to get to them now. But I'll start with one question. Can you translate regions of the consensus in SeqMan into protein sequence?
Sure, of course. If you click on your config so that you're in the alignment view, on the top left-hand side you have Translate and Consensus. So if you click on the triangle where it says Translate, you can see your top frame and your bottom frame. If you only want to see your top, for example, or your bottom, you can look at that here, and then it will give you the reading frame translation for your actual consensus file. And so that will be actually across the whole consensus file, not just individual areas.
Great, thanks. And the second question was, does SeqMan support next-gen sequencing data?
It does, yes. We have a separate program called SeqMan NGen that basically assembles next-gen sequence data. We work with most of the major sequencing technologies that are out there. If you're working with Ion Torrent, 454, Illumina data, we support all of those types of technologies. But when you assemble the data in a program called SeqMan NGen that we have, the assembled files are actually opened up in this SeqMan Pro software here in the Lasergene Core Suite. So you do the assemblies in SeqMan NGen, but all of the downstream analysis is done in this program here. All of your SNP detection, looking at actual sequence reads and your bases, all of that downstream analysis is done here. So if you know how to use SeqMan Pro, it's very easy to add on the SeqMan NGen software and get started with your next-gen data. And Matt Kaiser from DNASTAR will be giving a webinar in a couple of weeks to focus on those particular workflows.
Great. A couple of questions just came in. The first is, why would someone want to perform the sequence trimming that you showed? What's the reason for that?
Sure. A lot of times the beginning and the ends of your sequence are lower quality data than the middle sections. So it just kind of, the way the sequencers work, I think it kind of trails off in the beginning and the end. You may just want to do that because it's less quality data and it may not be as accurate as some of the other parts of your sequence. And you can always set that to turn that off, so you don't have to actually perform the trimming. If you want the entire sequence, you can do that and have that in the actual assembly.
Great, thanks. I think in the interest of time, we'll move on, but we'll address any other questions via email after the webinar. Great.
So move on. The last DNASTAR Lasergene Core Suite application that I want to talk about is the MegAlign software. Once again, this is the DNASTAR Navigator. This is just showing you an example of if you hover over any one of the modules, it will give you a description of what that particular module will perform for you, and the videos and tutorials that you have that go along with this Navigator section as well. But I'll click on MegAlign to launch the MegAlign software, and it opens up a blank window here. Actually the untitled sequence window. I'll just minimize this or maximize this window so you can see the whole sequence at the same time here. Really the main way to get the sequences into MegAlign, under the File menu option, you can choose Enter Sequences. And this will just give me a window where I can choose the particular protein or DNA sequence files that I want to import into the program. So if I click on the first one and then hold my Shift key down and click on the last one, it will actually highlight all of those sequences at one time. Click Add. I could have chosen Add All as well, and then Done. And this is basically giving me a window with the sequences on the left-hand side, my protein sequences, and then my actual amino acid sequences here, and my histogram and my alignment at the actual top. Now the main functionality in MegAlign comes under the alignment menu option. There's a variety of different alignment algorithms to choose from. You have a few major ones like the Jotunheim, Clustral V, and Clustral W method, and then you have a variety of one-pair alignment algorithms to choose from as well if you're only working with 2 sequences at a time. And that's the Wilbur-Lippmann, the Martinez-NW, and Lippmann-Pearson, and the dot plot method. So there's a variety of different algorithms to choose from when you're both aligning DNA and protein sequences. So in this example, I'm just going to choose Align by Clustral V. It performs a quick alignment, and at the top you can see a histogram of where the consensus sequence is similar to one another. You can see the more red and the more orange the sequences are, the more similar they are in those particular areas. Whereas the more blue or the more black those areas are, the more those actual sequences are different from one another. If I scroll down at the very bottom of my window, you can scroll through and actually see that the end of your sequence files, it kind of trails off. There's a lot of gaps in here, and this is quite different from the actual middle or the beginning of my sequence. So in this particular example, I want to go to my Edit menu option and choose Go to Position. I'll actually select a sequence range from 1 to about 415 bases. That's about where the time it starts to trail off at the end of my sequence and become a little bit more in disagreement with one another. And then from here I can choose Align, create alignment from selection, and that will actually ask me to save my new alignment that I'm creating. I'll save that on my desktop and just click OK. And what that's doing is it's basically just performing a new alignment and getting rid of the actual last bases that were a little bit in disagreement from one another. Another nice window that you have in the MegAlign software is under the View menu option, and I can choose Alignment Report. If you're using this data to actually publish or to present your software, to present your data with the software, this is a really great tool to do so. Under the Options menu option, you have the Alignment Report Contents, where you can show a variety of different things. You can show your consensus strength or your disagreement. You can show your translated sequences if you're working with DNA. You can break your alignment in a different number of residues, so it organizes it in a bit better fashion here. And you can copy and paste this information to Word or PowerPoint or to another program that you might want to use for a presentation or publication. The other thing that you can do here, I'm going to choose just my single amino acids. I think it's a little bit easier to look at versus the three-letter amino acid code. And then some of the other things that you can do is you can create decorations to this actual alignment report that you're working with. So I can hide the residues that match my consensus sequence, for example. And this is really just giving you a focus on where your differences are. And then if I create another decoration, I can shade my differences from my residues that differ from my consensus sequence. And I can choose a color and a level of shading, and that will actually show me those particular areas that are different from my consensus. And so this just gives it an easy way to point out where those differences or similarities are, and gives you a nice report to use, as I mentioned, for publication or even just presentations that you might be making. Some of the other views that are nice in MegAlign, you have the phylogenetic tree that I showed you in the very beginning of the PowerPoint presentation. You have a variety of different options that you can choose for your angled branches or your straight branches, and your cladograms that you can change here. You can zoom in and zoom out of the phylogenetic tree, and you can also perform bootstrapping algorithm as well to the phylogenetic tree, and it will give you those bootstrapping values on the branches of the tree. Another nice view that you have is the sequence distances view, where you're actually looking at the particular percent identity and the divergence of your actual sequences. And you can click on those in particular to see the actual percentages that you have here. One other example that I wanted to show you in MegAlign is just aligning 2 sequences at a time. So I'll close down these sequence windows that I have open. And if I choose File, Enter Sequences, I'll go to my Presentation Data folder and open up just a few different DNA sequence files that I have. I'll maximize this window that I have here. And in this case, I'm just going to highlight my sequences on the left-hand side. And then from the alignment menu option, you can see that my different one-pair alignment algorithms have become active. I'll choose the Wilbur-Lippmann. It will give me the different parameters that I can adjust, including the gap penalties. Click OK, and that will bring up the alignment view. Under the options, I'm actually going to choose the increase the font size so you can see this in a little bit more detail. And then you can change the colors. You can match the colors in green, for example, the mismatch is in red, and then you can change your consensus to a different color as well. You can show some vertical bars, show the sequence names, and make this a little bit more appealing so you can really see where the differences are of your sequences that you have. There's a lot of other tools directly in MegAlign that I really won't have time to cover today, but like I mentioned, for the other applications, feel free to explore the different menu options on your own, or go to our website to learn more on our training videos that we have on each particular application. So again, I'll open it up to questions, in particular on MegAlign, or any questions that you have about any of the Lasergene Core Suite applications.
It doesn't look like we have any questions at this time. If you can chat any questions in these last few minutes, that would be great. In the meantime, I will tell you a little bit about our next webinar. On June 27th, as Ryan mentioned, Matt Kaiser will be giving an overview of Lasergene Genomics Suite, which is our next-gen sequence assembly and alignment tool. And that's the same time on a Wednesday. If you have other ideas for webinars that you'd like to see presented, please contact me at webinars@dnastar.com or visit our webinars page to see a list of upcoming webinars, as well as a recording of this webinar. Okay, we do have a couple of questions. One that just came in, how do you know which alignment method is best to use for your project?
Sure, of course. And this is like I said, we have a variety of different alignment algorithms that we have. This is really where the help menu comes in a little bit handy. You can do a quick search either for that particular algorithm, or you can just go to the contents menu and choose, for example, performing a multiple alignment. Types of multiple alignments, it will actually list the different types that we have, the Jotunheim, Clustal V, and Clustal W, and it will just give you a little bit more information about when you might want to use that alignment algorithm, and it will give you the paper that it was published from, where you can even read up further on that particular algorithm. So it really depends on whether you're working with DNA or protein, whether your sequences have a lot of similarity or a lot of divergence. It really just depends on your situation. I'd encourage you just to look at the help menu here a little bit more to learn more about the actual algorithm that you might be wanting to use.
Great, thank you. And then a couple people have asked where this webinar can be found, and it's going to be on our webinars page in the next few days. Otherwise, the recording will be available on our YouTube channel, and you'll get an email after this event with the exact locations of those. So if you missed part of it, not to worry, we'll get you the recording in the next couple of days. And it looks like that's about all the questions. So if you have other questions, like I said, contact me or Ryan at ryanj@dnastar.com, and we hope to see you next time. Thanks for joining us today.
Thank you.